Hi guys, welcome back to the channel. In this video, I will be covering sex linkage, epistasis, pedigree charts, and chi-squared from the inheritance topic from the A-level biology curriculum. Now, I would highly advise you to watch my last two videos before watching this one. I will pop the link up now. Alright, so now looking at sex linkage. So X and Y are sex chromosomes. So out of the 23 pairs of chromosomes, one pair of chromosome is a sex chromosome, and we can have X and Y as our sex chromosomes. Now for our spec, only X chromosome can have a sex-linked gene. So how we showed genes before using a subscript, it's the same, but in this case we have X and Y chromosome. So only X chromosome can carry a gene. Let's say we had a gene for color blindness. Now the gene would only be on the X chromosome. We would not have a gene on the Y chromosome like this. The Y chromosome is just empty. We just put it there, but it would be only the X chromosome which would have a gene. Now because of this means that XY chromosome, so males in humans, but I've just put it in brackets because for example, in birds, XY chromosome is a female instead of a male, so it's flipped around, and this can trick some people out. But just remember that XY chromosome are more likely to show a sex-linked disorder. Now, this is simply because XY have only one chromosome, so that means they only have one allele for the gene. So, for example, if we were comparing a female to a male, so we have XX versus a XY. X-linked disorders are mostly recessive, so that's why for a female to have that sex-linked disorder, we would require both of the alleles to be lowercase, so both of them to be recessive. So as with the previous example, I'm just going to use the letter C. So we would have to have both lowercase c to have that condition. Now if we had a capital C, then the female would not have that recessive allele. But for a male, if we had even one C, now because that Y chromosome is not really involved in this, so if we had only one lowercase c, then the male would, or the XY chromosome would have that sex-linked disorder in that case. So the only way he will not have it is to have a capital C or a dominant C in that case. A very common exam question is, why are males more likely to have sex-linked disorders? And they might try and trick you. They might, for example, have a chicken, and they might say, why are female more likely to have sex-linked disorders than male? Then you can just simply say that recessive alleles will always be expressed in the XY chromosome. So first stating that they will have XY chromosome, and because of that they will only have one allele, and if it's that recessive allele for the sex-linked disorder, then that will always be expressed in that XY chromosome. Okay, so in recessive conditions, excess, so the XX chromosome can either be affected, not affected, or it can be a carrier. So for example, if we have a recessive condition, so that can either be lowercase c to be affected, or it can be a dominant C, uppercase C, and so dominant, and that will not be affected, or it can be carrier, so it can have one capital C and one lowercase c, so that it may not have the disease, it may not have the disorder, but it will be able to pass that disorder on to the offspring, so it's called a carrier. Alright, so just going through an example now. So it's told me that hemophilia is a recessive condition, and in this case the mother is a carrier for hemophilia, whereas the father is normal. So state the phenotypes. So in this case I'll be looking at my alleles for this. So it's told me mother is a carrier, and because hemophilia is a recessive condition, that must mean that the mother must be X. So in this case, because she's a carrier, I can say that this will be So I can say that would be capital A, and that can be lowercase a. So it's not really told me which letter I can use, so I can just use any I would like. So I've chosen an A for this case. So I've got capital A and a lowercase a because the mother is a carrier, and now because father is normal, that must mean that he will not have a lowercase a. So it must be X with a uppercase A and a Y which does not have anything on it. So if I was to cross them, so that would be X with a capital A, X with a capital A, and X, capital A again, and X with a lowercase a. Then I would have X, capital A, then Y, X, lowercase a, and Y. So it's asked me for the phenotypes. So in this case, to look at the females now. So from this, both of my female have a dominant A, so both of them will be unaffected. But I can see that one of them has a lowercase a. So in that case, this female will be a carrier. So her offspring may be able to have the condition for haemophilia. Now, looking at the males, looking at the first one, I can see that this is capital A, so he will not have haemophilia. But looking at the other one, he has a lowercase a, and the recessive allele will always be expressed. So in that case, that male will have haemophilia. Okay, so looking at another example. So in this example, they've told me that in a chicken, a male has chromosome XX and a female has chromosome XY. Now this is what I was on about. So in this case, the chromosomes are switched around for this chicken, or this happens for any bird. So we have, for a male we have XX, and a female we have an XY. So just watch out for this. So allele for a P-comb they've given me would be a capital A, which is dominant to allele for a single comb, which is lowercase a. So I can use them. So then it's told me that a farmer crossed a male with a single comb. So the male had a single comb and the female had a P-comb, and find the phenotypes of their offspring. So just writing down the gametes for this. So a farmer crossed a male with the single comb. So I know that male has XX in this case, so a single comb, so that would be lowercase a, and the female has a peacock, so that would be XY, and it would be an X capital A in this case. So just crossing them now, so I will have X capital A, X lowercase a, X capital A, X lowercase a, X lowercase a, Y, and X lowercase y. So in this case, looking at the phenotypes, so first looking at the phenotypes for the males in this case. So males, I can see that both of them have a capital A and a lowercase a. So this will mean that both of them will have a P-comb, because P-comb is dominant to a single comb, but both of them will be carriers for a single comb because we have a lowercase a in both of them. But looking at females now, so that's the XY chromosome for this question. So I can see both of them have a lowercase a. So both of them will have a single comb for this question. Alright, so looking at epistasis now. So epistasis is where we have interactions between two genes, where one gene will mask the expression of the other gene in the phenotype. So this is better explained with an example. Just going through an example, so they told me that in mice, melanin production, which is given a capital A, is dominant to non-melanin production, which is a lowercase a. And then they've told me that black coat color, which is now given a capital B, is dominant to the brown coat color, which is given a lowercase b. And finally, they've told me that if melanin isn't produced, then mice will have a white coat color. Right, so they've asked us a few questions. So first, they're asking us, what is the phenotype of capital A, capital A, capital B, capital B? Now, I know for my melanin production, I need capital A. My melanin production is the most important bit. So I have got capital A, so I know that I will not have a white mice because I have both capital A, so I will either have brown or black. So then from looking at the B gene now, so gene B, so I can see that both of them are capital B. So that means that I would be coding for black because that is my dominant for my gene B. So in that case, that would be black coat color. So that would be black coat. So looking at the second example, so I have a capital A, lowercase b, lowercase b. So again in this example, so I have a capital A, that's why I would be coding for melanin. So I can skip the white coat color and I can move on to the next bit. So I have lowercase b. Now I don't have a dominant B to code for black. So in this case, this will be recessive and this will be brown in this case. Next, I have a lowercase a, and a capital B, a capital EB. So if I was coding for melanin, so that would be black because I would have capital B, which is dominant and codes for a black coat color. But now because I have both lowercase a, this means I cannot code for melanin because I would require a dominant A, I would require an uppercase A to code for melanin. So this just means that I would not be able to have the black and I would have the white. Right, so looking at the next example, so I have capital A, lowercase a, capital B, lowercase b. So in this case, I will be coding for melanin because I have a capital A, and because I have a capital B, then this must mean that I would have black in this case. Now looking at the final example, I have lowercase a, lowercase b, lowercase b. Now if I was coding for melanin, I would then have brown. But because I'm not coding for melanin in this case, then I know that this must be white just by looking at a lowercase a. So I'm going to go through another example now. So in this case, they provided me with a table with the genotype and with their respective phenotypes, and then they've asked me to use the phenotypes and the genotypes to figure the phenotypes for the following. Try and do first is figure out what each of the genes does, so what the gene B does and what the gene E does. So looking at the genotypes and the phenotypes, I can see that I have yellow in two of them. And for the yellow one, I have a dominant B in one of them and both recessive in one of them. So in that case, I'd be thinking that B wouldn't really do anything to yellow because we have different alleles for this. But looking at the E genotype, the E gene, we have both lowercase e. So in this case, I would be thinking that lowercase e would be coding for yellow. And then I can try and figure out the rest from this. So in the other two examples, I do not have a lowercase e in any of them, but I do have the same e in both of them. So in that case, it shouldn't be the e, which should be affecting if they're black or chocolate. So then looking at the B gene now, so I have capital B, lowercase b, which is coding for black. So in this case, I know that if I have a capital B, I will be coding for black. And if I have a lowercase b, then that would be coding for, chocolate. So that would be chocolate. So it's a recessive, so I'll add another B in there. You can see from this is that if I have a lowercase e, this will mask everything else and I will have a yellow color. If we don't have a lowercase e, then we can have our other black and chocolate colors. So looking at the first example, so I have a capital B, and a capital E and a lowercase e. So in this case, we do not have two lowercase e, so it will not be yellow. And because it's capital B, this will be black for this example. Next, we have a lowercase b, and a capital E. So again, we will not have yellow because we do not have lowercase e, So in this case, this will be chocolate because this is recessive for gene B. Okay, so now looking at pedigree charts. So pedigree charts can be used to identify if someone in the family has a disorder and the probability of the offspring having that disorder. So mostly you will find that the squares will be males and the circles will be females, and when it's shaded, that will mean it's someone who has been affected by the disease, and if it's not shaded, then they have not been affected by the disease. Now looking at a few examples which can help us identify the trait of the disease. So in this case, both of our parents are affected, whereas one of our offspring is not affected. So in this case, we know that our disease must be dominant. Now this is because if our disease was recessive, then the offspring would be affected, because we would have, for example, let's say if the allele was CC, so it's lowercase c, then our offspring would also have lowercase c. But in this case, because it's not affected, then it must be a dominant disease. So in this case, both of the parents must be heterozygous. So for example, it might be capital C lowercase c, and this is why this could have been lowercase c, and that's why it's not affected. Now if both our parents are not affected, and if a offspring is affected, then we know that this must be a recessive trait. Because if this was a dominant trait, then at least one of the parents must have had the condition, but none of them have had the condition. So both of them, so these will be heterozygous, both of the parents, and then we will end up having a homozygous recessive in this case for the black to have the condition. Now finally, looking at this, so for example, the question might ask if how can you tell if this is a sex-linked trait? Now in this case, you can see that for this only the males are affected, and there's no females affected in this case, so that's what you can say. So looking at another example question, so it's told me that cystic fibrosis is a recessive condition, and it's given me a pedigree chart with the key. So the key is the same as before, and it's asked me what's the genotype for individual 6 first of all. So looking at the individual 6, I can see individual 6 is there, and I can see that the individual 6 is affected. Now because cystic fibrosis is a recessive condition, so individual 6 must have a recessive allele. So I can say lowercase c for this. Now looking at individual 2, so I can see that individual 2 is there. So individual 2 is not affected. So if it's not affected, individual 2, so it's a female in this case, so she can be either capital C, or capital C and lowercase c. Now to figure out if it's my capital C or if it's my capital C lowercase c, I would have to look at the offspring. So in the offspring, I can see that one of my offspring is affected by cystic fibrosis. So I know that so this was individual 6, which we have looked at, so they have the genotype lowercase c. Now to get lowercase c, my parent must be heterozygous because they are not affected, but they must be the carrier for the disease. So that's why I know that my individual 2 will have the genotype capital C lowercase c. Okay, so now it's asking me for individual 8. So again, looking at individual 8, I can see that one of the offspring is affected by cystic fibrosis, but the individual 8 itself is not affected by cystic fibrosis. So that's why, again, it would be capital C lowercase c. So the next question, it's asking me if couple 7 and 8 were going to have another child, find the probability of the child being affected. Now, I know that the individual 7 would have the same genotype as individual 8, because individual 7 isn't affected, but individual 7 must be a carrier because individual 10 is affected. Individual 7 must be a carrier just like individual 8. So for my gametes, I will have a capital C, lowercase c, capital C, and a lowercase c. So in this case, I'll have a capital C, capital C, capital C, lowercase c, capital C, lowercase c, and lowercase c, lowercase c. Now it's asked me for the probability of the child being affected. I only have one lowercase c, so that's why the probability would be 25% in this case. Okay, so you might be asked in an exam question why phenotypic ratios are different to the expected ratios. And for the reasons, you might be able to say that crossing over has occurred. And this is especially applicable for the autosomal linkage. So for autosomal linkage, always mention crossing over. Now for the second reason, you could say that fertilization is a random process. Any gamete could go with any gametes, so you might have a different ratio to what you expect. Thirdly, you could say that the sample size is small. Now, for example, so looking from the last example, so let's say we only have 2 offspring, and one of them has cystic fibrosis and the other one does not. So that will give us a 1-to-1 ratio. But this is because we have a sample of 2. Now, if we were to look at 1,000 children, then the ratio might be different. It would be different. So small sample size could be one of the reasons. And finally, epistasis. So remember, so epistasis means that one gene can mask the expression of another. So that could affect the phenotypic ratios as well. So just remember these 4. Finally, the chi-square test. So for inheritance, this is the most common, or is the only test that they can ask you for. So chi-square test is used to find if difference is significant between observed and expected. So if they ask you to choose a test and you choose chi-squared, and if they ask you for the reasoning, just say that we use a chi-squared because we want to find the if there's a difference, if there's a significant difference between the observed and expected. And you could also mention that the chi-square test is used because the data is categorical, because for most of inheritance, the phenotypes will be categorical, for example, looking at colors. So just mention these 2 reasonings. Now let's say you've got your calculated value and you've got your critical value. Then there's 2 ways of going from here. So if the calculated value could be more than the critical value, then you would reject the null hypothesis and you would state that difference between the observed and the expected is significant. And then you would say that probability of difference being due to chance is less than 5%. Now, if the calculated value was less than the critical value, then you would accept the null hypothesis and you would say that difference between the observed and expected is not significant, and probability of difference being due to chance is more than 5%. Now, there's more of this on my stats video, which I'll pop the link up for now, but just be aware of chi-square test for inheritance. Thank you so much for watching this video. If you did like this video, don't forget to subscribe to see more of these, and you can watch my recent videos by clicking on the links popping up. Thank you.